Hello everyone, this is Mr. Jackson, and today we're going to be starting part 1 of lesson 6, where we're going to be looking at RNA, short for ribonucleic acid, and then the process of how we make that RNA. And we have a specific name for that, and that is called transcription. So that's what we're going to be looking at today. We've got our learning objectives. You will know that you've mastered this lesson when you can describe how RNA is different from DNA. You should be able to explain how the cell makes RNA. So we're looking at that process of RNA synthesis. And it's right here with the next one, which is explaining how that process works and then being able to explain its significance. So why is this first step in your body being able to make RNA, which will later be used to make proteins, all the proteins in your body, why is that significant as a first step? Okay, so again, the lesson 6, the overall process that we're looking at is protein synthesis, how your body takes the instructions in DNA to make a polypeptide, which, remember, a polypeptide, you could look right here, that's just a protein's primary or initial structure, very, very basic structure. It's just a chain of amino acids, those individual building blocks or monomers of proteins, all in a nice line, all bonded together in a certain sequence. And it's important to note that these polypeptides can bind to other chains. And once they bind to other polypeptides, they can be folded into a three-dimensional structure. Those three-dimensional structures come together, and in this fourth level, really intricate structure, that is what we're actually talking about is a protein, right? So 2 or more polypeptides folded and bound together is what we actually would call a protein. And those proteins serve all of those important functions that we've previously talked about in our macromolecules unit. So a lot to thank proteins for, and just remembering that while they do have these 4 levels of structure, we, for this lesson, are going to specifically be focusing on just building out the primary structure, knowing that multiple primary structures can then come together to make proteins. Extremely, extremely, extremely important slide. The central dogma of genetics. You can also think about it as like a golden rule or a main idea of genetics. And this is going to include this process that you can see down here at the bottom. So this is the process by which we go from DNA, which we've had plenty of experience with in lesson 5. We're going to go forward into what we're talking about in lesson 6, part 1, which is RNA. And then we'll go forward again into part 2, where we look at the process of going from RNA to protein. These processes have very specific names. Going from DNA to RNA is called transcription. Easy way to remember this is we're making a small copy of some part of your DNA. It's kind of like if you go down to the guidance office and say, I would like a copy of my grades. Well, they're going to give you your transcript. And so if I take a small copy of only one portion of my DNA that I ultimately need to make a protein, then getting that copy or transcript of that information is called transcription. So that's the process that we're going to be looking at specifically in this part for now. But whenever we're referring to DNA, obviously we're talking about the genetic code, those instructions, and those are always going to be located safe and sound in your nucleus. Okay, so DNA does not leave the nucleus. It is far too bulky. It's large, so we can't get it out due to its size, but it's also, we don't want it out because we don't want to risk any sort of damage of that blueprint, that genetic code that we need to make any protein within any cell. So the issue here is that we're using this information to build all the proteins that we need in the cell and in our body, but proteins can't be made in the nucleus. And so they're going to be made outside of the nucleus, or in the surrounding cytoplasm, that fluid throughout the cell. And specifically, that's going to happen at those little protein factories that we call ribosomes. Ribosomes are outside the nucleus. Our DNA is inside the nucleus and won't leave. So we have to have a 2-step process. One part where we actually make a copy of the information we need, We create RNA, which is actually able, being a single strand and a lot more simple, we make this RNA which can leave the nucleus, and we use that RNA to then create a protein or a polypeptide chain at this ribosome. And so this image is one that I'm going to regularly go back to. It's a really, really good image for breaking down this process visually. But as we go through each part, we're really going to give you the opportunity to see the basics of how these processes work. So if this is still seeming like kind of intricate at this moment, that's fine, right? We're going to actually go into the details and make it a little more clear. So before we get into how we make RNA, we want to make sure that we understand the three main differences between RNA and DNA. Right, so the first one that you can see here is that the sugar in RNA is what gives it its name. It's going to be a ribose sugar, so we call it ribonucleic acid. Whereas for DNA, that backbone or that sugar in the backbone was deoxyribose. So the sugar has changed. Okay, RNA, as you can see here, it's a single strand of nucleotides, and over here for DNA it was double-stranded and those strands were twisted. For RNA, one key difference, and we're going to need this moving forward anytime we're making copies, we're moving on past DNA replication, and so now rather than thinking about the bases that are exclusive to DNA, which are your G's and your C's, those A's and your T's, RNA doesn't utilize thymine. You can think of it easily as thymine doesn't travel well. So instead of A binding with T or A bringing in T's, whenever we're making RNA, we're going to replace those T's with U's. RNA uses uracil. And again, you can think of it as being just a little more travel friendly. We got three main types of RNA, and I'll let you know as we go which types are serving what purposes. But now, just generally, we've got our messenger RNA. This is basically going to be the copies that we make from the DNA inside the nucleus. Okay, these are going to be the traveling messages. So they carry copies of the instructions in DNA. These instructions are for assembling proteins, and it goes from the nucleus to the ribosomes outside of the nucleus in the cytoplasm. These ribosomes are where we actually do the protein synthesis. So that's messenger RNA, the mobile form. Transfer RNA, it's also mobile, happens to bind to and carry amino acids. This would be an example of how it binds to and carries an amino acid here at the top. And then it's going to carry that to the ribosome and use these three, what we would call bases or an anticodon. We'll get to that in part 2. But all we got to know is that this serves as a little puzzle piece that we can use to basically match an amino acid to some portion of our messenger RNA. And then finally, you have the ribosomal RNA. It's called ribosomal because this specific type of RNA makes up all of your ribosomes, which are your protein factories. So we use this messenger RNA to assemble chains of amino acids at the ribosome, which is made up of mostly this type. For the rest of our conversation, we'll understand that that type of ribosomal RNA is there, but mainly we're talking about messenger RNA in transcription and transfer RNA in part 2 with translation. All right, so some basics. Transcription, this process in which we go from DNA to messenger RNA. Okay, so another way to think of transcription is RNA synthesis, is how we make RNA. The purpose of this is to be able to carry the code or the instructions from the DNA, right? So the specific instructions for only a specific protein, we make a small copy of exactly that info so that we can carry it outside the nucleus because the DNA can't leave. And then the proteins would ultimately be made in the cytoplasm. For transcription, please understand that this is happening where the DNA is, inside the nucleus, and we start with DNA and we end with messenger RNA. And we're going to go into this process. So what does this look like? It's going to start by, we're going to, just like with DNA replication, we are going to unzip the gene, right? Remember, that's only a small section of your DNA code. So we're going to unzip a gene of information, and that gene is going to be specific to building a specific protein that the cell needs. So we unzip this gene that we need to copy, and we expose these bases. We're going to then use those same complementary base pairing rules to match RNA bases with the exposed DNA nucleotides. Now remember, if you can see here, DNA uses A's with T's, right? So A's with T's, C's with G's. RNA does not use thymine. It doesn't have the T's. Instead, we use uracil, and that's in the U. So now we can pair up anytime if I'm making mRNA, if I'm making an RNA strand, anytime I see an A, I'm going to bring in a U, which is that uracil. And anytime I have a G, I still bring a C. So you can see how that process might work, right? Where I've exposed these bases by unzipping, and then I can use those same base pairing rules, A's and U's, C's and G's. After we make all of the RNA strand, if we've copied all of that specific gene, we can then release that completed mRNA, that mobile messenger RNA. And then after that's released, and we've done our job, we've made the copy, then we can zip back up this gene, this part of the DNA, and the messenger RNA can then leave the nucleus to go into the cytoplasm and deliver the message. So another way of seeing this here is in transcription, segments of DNA, we call those genes, information for a specific protein. These segments serve as templates, right? Blueprints to produce complementary strands of messenger RNA. That is the name of the game. So if we have our initial DNA molecule, we would unzip it in the middle. So we have the left and the right original DNA strands. This left portion is going to be a gene or a section of DNA that I need, instructions for making a certain protein. I would then do the process of transcription to create a messenger RNA here in the middle. And then I can release, once I'm done making that copy, I can release the messenger RNA and the DNA will zip back together, and I've done my job. I have this message ready to deliver. And so we're going to finish up here with a small example. We are going to have a couple of practice activities and some escape room challenges and just a lot of practice with utilizing the transcription and translation process. But I want to make sure it's clear how we go through this process. The same way we just did on the previous slides, you're going to have plenty of opportunities to do this yourself. So if I were to give you the following DNA sequence, for every T that I give you in DNA, it can still bring in an A for RNA. RNAs use A's. Every A in DNA is going to bring a U because remember, messenger RNA, we don't utilize thymine. Doesn't travel well. Easy way to think of it. For every C, we bring a G. Every G, we bring a C. These base pairing rules have not changed. Only one of the letters has. A's and U's, C's and G's, and vice versa. So if I were to give you a DNA strand here in blue, and I say, bring me the mRNA that's complementary, this is what this should look like. T becomes an A becomes a U, C becomes a G, G becomes a C, and so on and so forth, until we have that full copy of that specific gene, which are those instructions, again, for creating the proteins that we need. And so at this point, we're going to close out part one. And remember, this was all about RNA, its structure, how it's different from DNA, and then how we actually create that RNA through the process of transcription. And so we're going to call it there with part one, and we'll see you at part two. And I hope you found this helpful.